it's concerning because the more the virus has the opportunity to do that, and the more we see the virus do that, it's an indication that the virus is moving closer and closer to, again, a virus that can easily transmit from person to person. And once we've hit that threshold, again, an H5 virus that transmits easily from person to person, we're in a pandemic. And then it is, 2020 all over again, maybe worse.
Hi, thanks for tuning in again to the Doctor Podcast Show. And this is episode number 83. And I'm Dr. Robert Seichert, your host today. Today, we're talking about a very important topic that's been in the news a lot lately, and I think maybe in the news a lot more in the coming months in 2025. And that topic is bird flu, which unfortunately there are more and more cases of it in animals and humans in the USA. And there's a possibility that it could possibly become a pandemic. So we're going to discuss that today with one of the world's top experts on bird flu. And our guest today is Dr. James Lawler, who is at the University of Nebraska Medical Center. He's a professor in the Division of Infectious Diseases, and he's board certified in both infectious diseases and internal medicine. He also has a Master of Public Health degree and diploma in tropical medicine and traveler's health. He's currently the director of the International Programs and Innovation at the Global Center for Health Security at the University of Nebraska, and we'll talk about that some more in a few minutes. He was a Navy commander, and thank you very much for your service. And he was a chief of clinical biodefense research at the Naval Medical Research Center in Fort Detrick, Maryland, which we'll talk about some more as well. He was also an attending physician at Walter Reed National Military Medical Center and served several White House assignments as a Homeland Security Council and National Security Council staff, where he worked on biodefense, pandemic response, and health preparedness. He treated Ebola patients in Western Africa during the epidemic there about 10 years ago. And he's also trained the Department of Defense medical personnel working with infectious disease patients. So all of these credentials are critically important for our discussion today. Thanks very much, Dr. Lawler, for taking the time today to join us to talk about this important topic. We appreciate it.
Of course, thanks for having me on.
So I want to start by asking you, how did you become interested in infectious diseases after being board certified in internal medicine?
So I went into my residency in internal medicine thinking that infectious diseases was probably my first choice. I briefly flirted with the idea of doing cardiology, and much to my financial advisor's dismay, I chose ID instead. But I'd always been interested in infectious diseases. Even in medical school, my favorite lectures were from our pathologists who specialized in tropical diseases. And in the military, I was in the Navy during my residency training and for 21 years. And the military is a really great place to practice infectious disease, particularly if you're interested in tropical medicine or emerging infections. And so it just turned out to be a great fit.
Wow, sounds great. It's interesting. I'm an ophthalmologist, as you know, but my second choice, and I almost did it, I went into infectious diseases because I enjoyed that subject as well. I felt like it was always a war and a battle between me and some vicious bacteria or virus, and I would win. But the last moment, I changed my mind and went into ophthalmology. Now, what was it like working at Fort Detrick Naval Medical Research Center in Maryland? Were you there during the biowarfare era where they were working on creating nasty viruses and bacteria? Or were you there in the biodefense era where they were working on how to treat those conditions or prevent them?
Well, the offensive biological weapons program was a bit before my time. So that program was shut down in 1969 with an executive order from President Nixon, actually.
Hmm.
So from 1969 onwards, the U.S. did not have an offensive program for biological weapons, and all of the work done at Fort Detrick was for biodefense purposes. And for your trivia fact for the day, a large proportion of Fort Detrick is actually part of the National Cancer Institute. There's
Oh,
a large research component of NCI that is up at Fort Detrick. And the reason it's there is to actually provide transparency and oversight to assure people that the offensive program indeed was shut down. So they thought having a bunch of cancer researchers up there on site would give people confidence that there was at least somebody, peeking behind the curtains to make sure that folks up there still weren't doing bad things with bugs.
right. Wow, I didn't know that. That's actually quite interesting having somebody checking on you and make sure you're doing the right thing. Now, Fort Detrick is a biosafety level 4 facility. Can you explain what that means and how many facilities are there like that in the USA?
Right. There's actually three facilities up at Fort Detrick that are now biosafety level 4 laboratories. So I had a couple of stints up at Fort Detrick. I first was up there as a fellow doing research projects starting in 2002 at USAMRIID, which is the U.S. Army Medical Research Institute for Infectious Diseases. USAMRIID is kind of the prototypical biodefense laboratory that people think about at Fort Detrick. And part of USAMRIID has BSL-4 laboratories. And over the course of my time at USAMRIID, I was eventually stationed there full time from 2004 till 2006. And during that time, actually got qualified in doing BSL-4 research. And the biosafety level categories are just standards or guidelines for biosafety practices and construction of the laboratory itself to ensure that you have adequate containment of pathogens. Biosafety level 1 and 2 laboratories are laboratories that people would commonly encounter in a high school biology lab would essentially be a BSL-1 laboratory, and BSL-2 labs are generally hospital clinical microbiology labs. When you get to BSL-3, there are additional standards that are built into the engineering of the building itself, negative pressure and controlled airflow that's HEPA filtered, access control, special procedures, and personal protective equipment that you wear. And then BSL-4 is the highest level. There's actually two different types of BSL-4. One is essentially a cabinet laboratory, so a biosafety cabinet or a biocontainment hood, which has the gloves that you see in kind of the old Dr. No movies. That is technically a BSL-4. But when people think of BSL-4 labs, they usually think of the spacesuit containment laboratory. So where the personnel actually go inside the hot zone or high level isolation space, and we're wearing spacesuit looking equipment that is usually tethered with air supply, a positive pressure air supply from a hose that you hook into your suit. So there are three of those laboratories at Fort Detrick. USAMRIID, then there's a Department of Homeland Security laboratory, the NBAC, and finally NIH built a laboratory up there where I also worked a little bit later in my career. And across the US, there's, I think, five more BSL-4 laboratories with the spacesuit setup.
I know the CDC has one, and there are a few other
Yes. Yep.
facilities.
CDC has one. There's the Needle in Boston. The NIAID, National Institute for Allergy and Infectious Disease, has a second integrated research facility, BSL-4 lab in Montana. There's a BSL-4 laboratory in Galveston, Texas, and then there's one in San Antonio, Texas.
Now, these labs contain like the nastiest, most vicious and deadly bacteria and viruses that exist on the planet. Is that correct?
True. For BSL-4, all of the pathogens are generally viral because they are diseases for which we have no available or limited available countermeasures like vaccines or antiviral therapies. They are also diseases that are transmissible to people and are diseases that generally have a high lethality. And so for most BSL-4 pathogens are things like Ebola or Marburg or Crimean Congo hemorrhagic fever, all of those nasty hemorrhagic fever viruses that we think about, a couple of other encephalitis viruses like Nipah and Hendra, and things like smallpox. For instance, when smallpox is worked with in the laboratory at CDC, it is in BSL-4. So pathogens that have a massive consequence for human to human transmission if they were to circulate again.
Were there ever any leaks while you were there, or were you nervous working in those type of facilities?
Well, I wasn't nervous. I think all of us who worked in BSL-4 and people who still work in BSL-4 are cautious and very detail oriented. There actually has not been in the U.S. a documented infection, an occupational infection, or known uh, exposure, a significant exposure to a BSL-4 pathogen that's resulted in infection. And um, I think people are a little bit surprised to hear that I am much more worried about BSL-3 pathogens because
Hmm.
there are many more BSL-3 laboratories, and the level of precautions that are built into BSL-3 processes are such that there's just more opportunity for error and occupational exposure. And so the BSL-4 laboratories, when they're run well and appropriately, which is obviously the big part of the equation, I think that the risk is very manageable. And obviously, we have to do research on these pathogens to be able to develop new countermeasures. That's why we have now vaccines for Ebola that have been shown to be effective, at least in the clinical trials that have been done in Africa. And we now have some therapeutics against diseases like Ebola and Marburg, and we would not have those if we didn't have the ability to do BSL-4 laboratory research.
That's interesting. I never considered that the BSL-3s are actually more risky because the safety measures are probably not as high, but hopefully nothing escapes from there. And we also have bacteria like anthrax, right? It's deadly.
Yes. Yep. So anthrax would generally be in a BSL-3 laboratory. Other diseases, tularemia, for instance, is worked with in a BSL-3. So a lot of the bacterial pathogens that we consider special agents and dangerous are in BSL-3. And it's also the case that some of those pathogens like tularemia or Coxiella, the agent that causes Q fever, can be infectious in very, very small doses, as few as one bacterium from Coxiella can potentially infect a person. And so that's another reason why some of these BSL-3 pathogens are, in many ways more concerning to me. But also they don't necessarily have the ability to cause massive person-to-person transmission outbreaks.
Wow. One bacteria. That's pretty frightening. Now, the lab in the Wuhan laboratory where there's rumors that the COVID virus came from, that was a BSL-4 facility, I believe, but apparently wasn't really kept up and may have been not as safe as everyone thought. Is that right?
Well, the Wuhan does have a BSL-4 laboratory, but the laboratories where the Wuhan Institute of Virology was doing research on bat coronaviruses was actually not a BSL-4 laboratory. That work was being done in BSL-3 and potentially BSL-2 laboratories. And so if there was, if the origin of the COVID pandemic was an escape from one of those laboratories, it wasn't an escape from a BSL-4, it was likely an escape from BSL-3 or BSL-2.
Hmm. That's interesting. Now, what was it like working at the White House and Department of Homeland Security regarding these diseases and pandemics?
So I did not actually work at the Department of Homeland Security, DHS. I did work on the Homeland Security Council at the White House. So after 9/11, there was a new entity that was stood up within the White House to address the homeland and terrorism threat, analogous to the National Security Council. And that was the Homeland Security Council, which existed throughout the Bush administration. The Obama administration actually folded HSC into the NSC. And so when I was at the White House the second time during the Obama administration, that office had been folded into NSC. So I worked in the National Security Council my second stint at the White House. But it was an interesting place to work, but honestly, some of the most enjoyable years of my career just because of the people I had the opportunity to work with. I really like to think of the group of folks that I worked with as the dream team. I've never been around a collection of people as accomplished and smart and clever as the group that I had the good fortune to work with in both of my tours at the White House.
Wow, that's great. An awesome experience. Now, I mentioned earlier you were also in Africa during the Ebola outbreak. That must have been also very frightening, and that's why I went into ophthalmology, I guess. What was that like?
Well, again, I wouldn't uh, necessarily say frightening, although there were some circumstances, but that was also what I had trained to do for a long, long time, both in working in biocontainment in laboratories and also eventually doing work in outbreak response in Uganda a couple of years before that. And so the, to me, it was more like kind of the game time that we had been practicing for a long time. And obviously, there were a lot of issues in West Africa, especially early in the response. And we eventually learned a lot about how to care for these patients more effectively, how to do isolation care better. But it certainly was an interesting time.
Right. So tell us about what you're doing now at the University of Nebraska Medical Center, and your role there.
Sure. So I left the military in late 2017 and was looking for a place to land afterwards. I wasn't really sure where I wanted to go. Actually, uh, didn't think I wanted to go to an academic medical facility, but I gotten to know the folks here at the University of Nebraska Medical Center for during the Ebola outbreak in 2014, given that the Nebraska Biocontainment Unit here was one of the three facilities where American patients were repatriated with Ebola from West Africa. So this team was just amazingly accomplished and did great work in treating Ebola patients during the outbreak, but also in publishing their protocols and their approaches and going out on the road to train other hospitals to be able to manage these types of patients. And so I was really impressed with what they did in 2014, and then had the opportunity to work with folks here on a few projects in Africa, training folks on isolation care and also doing some research on emerging infections in West and East Africa. So the more I got to work with folks from the team, the more impressed I was. And eventually we got to talking about what might happen after I got out of the military. And about that time, the university was standing up this Global Center for Health Security, which is the group that I belong with now. And this group really brings together all of the different research and training and operational elements that exist here at UNMC and kind of coordinate all of this. And I think really has created a unique center that is different from any other that focuses on health security. So we tend to be much more practical in how we look at the health security challenge and focus on healthcare systems and healthcare delivery and bringing practical solutions to health security challenges. So we like to say that many health security institutions out there are are very powerful think tanks, but we're a do tank, and that's the attitude that we like to take.
Right. So are you affiliated with the government in some way, or is this just the University of Nebraska?
So the University of Nebraska Medical Center is a public institution. We are the state medical school and medical education campus for the University of Nebraska system. So we are a state government entity, but as with all universities, we maintain as much independence as we can.
So I want to go to bird flu now, which I'm very interested in and have been following very closely in the last year. How is bird flu different than the standard seasonal flu that we see every fall and winter in the USA and that we get vaccinated for? And what's the concern with bird flu?
Wow, how much time do we have? No.
We can go to midnight.
Great question. So it turns out that all sorts of different animals get flu, and not just people. So birds get flu. Almost every mammal gets some type of flu. Even whales and dolphins get flu. And so influenza viruses are pretty common among different species of animals. And different influenza viruses tend to have some specificity in terms of what animal species they infect based on the the little proteins that stick out of the virus that we call hemagglutinin and neuraminidase are the two main proteins that are on the outside of the virus, and the hemagglutinin is the one that really is responsible for binding to a host cell and gaining entry. And so the influenza A viruses that are the main pathogens for humans to cause seasonal flu. We also get infected with a couple of other viruses. The only other clinically important virus is flu B. But influenza A is the one that causes the most severe disease and the most common infections in humans. It turns out that, again, influenza A viruses infect lots of different animals, including birds. And so the issue becomes when flu viruses jump back and forth between species, there's a potential for a flu virus to be able to infect humans that humans haven't seen before. Right. So I mentioned those different H and N subtypes that stick out from the surface of the flu virus. Our immune systems are very well attuned to recognizing and fighting off to some extent flu viruses that have circulated in humans before and that we've seen before. So currently, our seasonal influenza viruses that have been circulating really since 1977 have been both H3N2 and H1N1 viruses. We had a bit of a switch from that old H1N1 to a new H1N1 in 2009 associated with the 2009 pandemic. But essentially, those are the viruses that humans have seen really for almost the last 50 years. So a new virus showing up that maybe would be an H5 virus, right, that's now circulating in birds, would be a virus that humans have not seen before. And therefore, we would have very limited population immunity and individual immunity against one of those viruses. And so that's why a new influenza virus like that is concerning. There's also differences in what type of cell receptors these viruses like to bind to. So bird viruses tend to bind to a certain conformation of the sugars on the outside of our cells. And that conformation, which is common in birds, is seen only deep in the lungs of humans. A different conformation is seen, which we call alpha-2,6, in the upper respiratory nasal mucosa for humans. And those are the viruses that humans can transmit effectively from person to person because you shed lots of virus from the upper airways like that. And so again, when a bird virus mutates to all of a sudden be able to bind to that alpha-2,6 receptor that we have in the upper airways, again, that becomes a virus that can potentially pass easily from person to person, but still may maintain some of the characteristics of the bird virus that may make it dangerous. And so since the early 2000s, there have been almost 1,000 human infections documented with H5N1. And the mortality rate from those infections has been about 50%. So even though there's been millions and millions and millions of birds infected and probably lots of people exposed, infection has thankfully been rare. But when those human infections do occur, they've been quite severe. And so that is why many scientists and experts are concerned about this H5N1 virus that's circulating around. It potentially has an opportunity to, move into humans, become more adept at transmitting person to person, and that would be a dangerous virus with certainly high pandemic potential. I think if we had an H5 virus that became adept at passing from person to person, it would almost certainly become a pandemic.
And it sounds like it's more lethal than COVID, right? Nearly
Well,
50% mortality.
sure, and that is certainly an overestimate, just because we don't probably catch a lot of the less severe infections that have occurred in many areas where these infections have been more common, Southeast Asia, Egypt, and so that denominator is probably much larger, and the true case fatality ratio is probably a lot lower than 50%. But even if it were 5%, right, 10 times lower, that is still twice as lethal as the 1918 pandemic H1N1 virus. And so
Hmm.
um, there's a long way to go till you get down to a level of lethality that would not result in a catastrophic pandemic. And the reason that pandemics are uh, so scary and so impactful is not necessarily the high lethality. And we saw that with COVID, right? SARS-CoV-2, in terms of lethality on an individual scale, probably was 0.5%, for all comers in 2020. But the problem is when 30 to 50% of the population gets infected, when you have 330 Americans or 7 billion people in the world, all of a sudden 0.5% becomes a huge number, as we
Right.
saw. So that's the concern, is not only the lethality, but, and again, I think that the true lethality is probably much lower, and we're seeing some of that now in the human cases we've seen in the US. But again, even if the case fatality ratio is 0.5% or 1%, in a pandemic where 30 to 50% of people are infected, that becomes a huge number that overwhelms our healthcare system, as we saw in 2020.
Right. Now, when was the first case reported and where was it reported? Do you know where that might be?
The first case of avian influenza, all comers, or the first case in this particular
No,
outbreak?
all comers. I think it goes back
Yeah.
a couple of decades or so.
Yeah, yes. Avian influenza has been around for many decades. I mean, avian influenza has probably been around for thousands of years, but we've
Oh,
known about it for many decades, and have been tracking H5N1, highly pathogenic avian influenza, for more than 30 years.
30 years.
And so there are multiple different H and N combinations that can be seen in bird flu, H7N1, H9N2. H5N1 has been the dominant avian influenza virus, causing highly pathogenic avian influenza for the last couple of decades, and it really took off in the early 2000s. So there was an outbreak in 1997 that was relatively large, and then and this was all in Asia, and then again in the early 2000s, another large outbreak, and that's really been the dominant highly pathogenic avian influenza that's circulated across the globe.
So how has H5N1 spread around the world? Is this just from migrating birds, basically?
Yes, it infects primarily wild birds and a number of different species of wild birds, although obviously when it gets into domesticated poultry, it causes huge problems. And the wild bird populations that it can infect are things like cranes and swans and other birds that have very long migratory routes. And so it creates a a great shuttling mechanism for the virus to move across the globe, and scientists have been able to follow the progress of the virus moving through these long migration routes. And that's generally how domesticated poultry and probably how cows in the US became infected with the virus, with an introduction from migratory birds. And that's also happened in marine mammals and lots of different species of mammals over the last few years. And again, that's why this particular H5 virus that we've seen since 2022, which is called a clade 2344B, has been especially concerning because we've seen infections in a large number of mammalian species, which we have not seen before. And we've seen transmission from mammal to mammal again, which is something that we had not seen before. We've seen high levels of fatality in mammals. And obviously now in the US, we're seeing this huge outbreak that's affecting dairy cows.
Right.
And all of these things are new and unprecedented. And when a dangerous virus does new and unprecedented things, we should be very concerned.
Right. Yeah, I've read down in South America there are seals and walruses dying from it. Cats have been infected,
Yeah,
ferrets, and other animals.
yeah,
So
huge
what
die-off.
happens is a sick wild bird happens to land in a chicken farm somewhere and infects all
Right.
the chickens.
Right, that's probably what happens. Or, a migratory bird flying over poops in the chicken's water source or food source, and that's how the chickens get infected. And once a chicken's infected, it spreads rapidly through a domesticated flock.
So how did the cows get this, the dairy cows? From what I've read, there are dairy cows infected in about 15 different states in the USA, mostly in the Midwest. But in the last few months, there have been a few hundred herds infected in California as well. How do you explain that these large herds of dairy cows are getting infected at the same time, yet they're geographically dozens or hundreds of miles apart?
Yeah, so it's been really interesting, and I've certainly learned a lot about dairy animal husbandry and practices over the last several months. So what we know is there was probably one single introduction from a wild bird into dairy cows, most likely in the panhandle of Texas, that started the outbreak. But then the transmission that's occurred since then has actually been from cow to cow. And it turns out that in the dairy industry, moving cows around from state to state is actually very
Hmm.
common practice. And so there is, on a daily basis, transportation of cows from herd to herd and state to state. And then these cows are able to infect each other in a herd so that a large proportion of cows within a herd become infected. It's also true that a lot of people and equipment are moving back and forth between different farms and locations. Sometimes dairy workers may work in multiple farms, and certainly the trucks that come and service them and vets that take care of the animals are obviously working in multiple farms. And we know that cows that are infected are shedding enormous amounts of virus in milk. And so that milk is highly infectious. And it's probably how cows are passing the virus from one to another through the milk, and obviously why it seems to be affecting dairy cows mostly because of the high levels of virus that are seen in the milk.
So now the California herds, however, are very far away from all the other Midwest and Texas herds that have been infected. How do you explain that?
I think that it is quite likely again that cows were transported into California from one of the other states where outbreaks were occurring. Again, this movement of cows between states and herds is quite common. Turns out California is actually the largest dairy state in the country. They have the highest number of herds and dairy cows of any state. So not surprising that eventually it would get into California. I think California's done a bit more in terms of surveillance and detection. And so that have detected more herds that have been infected in California than in any other state. Again, I think partly because of the fact that they have more herds in other states, but also again because I think California has been a bit more proactive in trying to actually find infection. The strategy of most states, unfortunately, and nationally, our strategy has been to bury our head in the sand.
Hmm. I was just going to ask you, it would seem to me like the government, I guess the Department of Agriculture, should have banned interstate transfer of dairy cows once the first outbreak occurred.
Well, they eventually did implement a rule that cows being moved between states had to have a test, a negative test to be able to move. The reality is there's no mechanism to enforce that and to oversee that. So it's, I think, questionable whether or how many cows that are moved across state lines now are actually getting a real test. And
We're talking
so,
about thousands and thousands of cows, probably
yes,
very
yeah,
difficult to
exactly.
test them all and then
Yeah,
keep track of who's positive. And
right.
the
And
time you
so
get the result, it's maybe too late.
without an enforcement or tracking mechanism, it becomes very difficult to make sure that those cows aren't safe to move into a new herd.
Now, I know the chicken and poultry farms, they've killed tens of millions of chickens who were infected just to get them out of the system so they don't transmit it. What do they do with a sick cow that has bird flu?
So the cows do become ill. They do have a drop in milk production, and the milk that they produce apparently is um, looks different. And so those cows are isolated away from the other cows on the farm, and the milk is supposed to be discarded. But these cows, they still have to be milked every day. If you've ever had an animal that is a milk producer, we had goats on our farm in Maryland, right? You can't just not milk those animals. They have to be milked, or else they potentially get mastitis and other significant complications. And so that those cows that are actively ill and we know have lots of virus in that milk are still getting milked, and thus the risk of spreading continues. The other piece of these cows is that many cows who are still shedding large amounts of virus, either before they develop symptoms or maybe they don't develop symptoms as severe as many of the other cows, or they may persist in shedding virus, um, also can be shedding large amounts of virus. So it's not just the cows that look physically ill that are shedding virus in the milk. Other cows that look relatively healthy can also be shedding. And so the virus is unfortunately getting into a large proportion of the U.S. milk supply. Um,
Hmm. Seems like
Fortunately,
almost an impossible task to separate cows that have the virus from the ones that don't, because you're using the same milking machines, the
right.
same facilities. It's probably impossible to sterilize that equipment. So it
A
seems,
difficult challenge to be sure, right? But one that would require um, changing workflows and practices and also testing, right? So you're not going to be able to do any of that if you're not proactively testing on these facilities. Yes, it would be challenging. Some of these large dairy facilities have over 1,000 cows that are milked every day, twice a day, and so that is yeah, not an easy thing to do, but also um, doing nothing is not necessarily a good idea either.
right. Now, so the milk or cheese products that come from these cows are basically teeming with the bird flu virus, with H5N1. And the recommendation has been not to drink raw milk or use cheese made from raw milk for that reason, because you can get sick from that. Is that right?
Well, The pasteurization process, the standard pasteurization processes that are used in the dairy industry are sufficient to kill the virus and to make it safe for consumption. So pasteurized products are safe to consume, right? At least have a very, very, very low risk because the studies that have been done so far indicate that the virus isn't inactivated by those processes. But raw dairy products that are not pasteurized likely contain a large amount of H5N1 virus, and certainly from herds that are affected. And in general, let's back up and say raw dairy products in general are not safe to consume. There are all sorts of nasty bacteria that are passed in raw milk, from Salmonella to E. coli to Campylobacter, you name it. Lots of very bad infections can be transmitted through raw milk. Sadly, we just had a case not too long ago in the children's hospital of a kid who had renal failure from an infection transmitted from raw milk that the family had consumed. So these things happen. They happen relatively frequently, but if you needed another reason not to consume raw dairy products, maybe this could be another one, because it is high risk for sure for
Right.
transmitting H5N1.
Right. So I read about 10 days ago, California found raw milk that was infected in about 10 different retail stores. People were buying raw milk. It was eventually recalled. I don't think there have been any cases reported of human infections from that, but shouldn't the government say raw milk is forbidden, can't sell it?
Well, there's a couple of different things that I think come into play there. First of all, the federal government actually does not have jurisdiction to limit the sale or consumption of raw milk unless it crosses state lines, right? So the FDA, which regulates milk, uh, only has authority under the Interstate Commerce Act, right? So things that don't cross state lines don't come under FDA jurisdiction, and therefore states are the ones that actually regulate the sale and consumption of raw dairy products. There are some states in which it is illegal, but many states do allow the sale of raw milk. And again, it's kind of one of those individual rights things, and perhaps at some point people have to be able to take their own risks. People smoke, right? And that's terrible for you as well. Um,
Alcohol.
yeah, but I do think that certainly when you start giving it to kids and giving them life-threatening infections, that is certainly a concern. And again, there's no proven scientific benefit to consuming milk that hasn't been pasteurized, right? Pasteurization does not uh, alter the milk in important ways that diminish its nutritional value. And there's never been any studies that show that there's any benefits from raw milk above pasteurized. You just buy all of the risk of the pathogens that are in there. So we can
So
think
I
about individual
understand.
rights and those things, but
So at least there should be a label on that refrigerator in the grocery store in California that says raw milk is hazardous to your health, just like they put the message on the cigarettes, because there's probably people who
sure.
just don't know.
Well, there are a lot of people who really do believe that there are benefits to consuming raw milk, and that's just not again not the case, has not been proven in any way. But we have warning labels on cigarettes and people still smoke. So
Right.
I'm not sure that that's going to solve the problem. But again, at some point, you take your own health into your hands. But yeah, it is not a good idea to consume raw dairy products.
Right. Can you explain what pasteurization means? What's that process and how does it kill the viruses and bacteria?
Yeah, so simply put, pasteurization is just heating up the milk. We've known about pasteurization since Louis Pasteur, right? That's how we got the name. It's been around for 140 years. It works. It's very effective. It does not, again, affect the nutritional value of the milk. And there's different ways to pasteurize. There's heating it up to very high temperatures, below boiling, but high temperatures for short periods of time. More standard processes are kind of a slow heating process, that heats it up just enough to kill the bacteria, but and viruses, but not enough to really significantly alter the milk proteins themselves. And so um, that there, people who drink raw milk, I've never had raw milk, but say that it tastes differently. It may, but what tastes really bad is the uh, endotracheal tube that they put down your throat when you're in the ICU.
Right. Don't eat raw dairy products. Now,
Exactly.
in the last year or so, there have been about 55 cases of humans in the USA who have caught the H5N1 bird flu from either poultry or cows. How did that happen?
Yes, I think we may be up to 57 now. There's new cases
50.
popping up almost every day now, which is scary. The majority of these cases have come from exposure to dairy animals, to cows, in dairy parlors. And there was a study that was just published from the CDC looking at dairy workers on farms with documented infections in Michigan and I think Colorado. And they showed that about 7% of the dairy workers had antibodies against H5N1 virus. And so probably have been a lot more than 57 infections nationwide. These are just the ones that we've discovered. And again, because we're not really testing very aggressively. But as I mentioned, the amount of virus in the milk in these animals is really sky high. And in those dairy parlors, they are hooking up these cows to giant sucking machines that generate aerosols by themselves. And then after the milking is finished, they're spraying down the equipment and the
Mm,
milking parlor itself with a high pressure hose. And so you're creating all sorts of splatter and droplets and aerosols that contain virus. So it's actually not surprising at all that humans are being infected. There have also been some infections that have come in poultry workers, mostly poultry workers who have been involved in culling activities, right? So euthanizing birds and flocks that are infected, and those culling operations are messy. It's usually in a hot, humid, crowded uh, poultry barn, and so lots of opportunity for people to be exposed there. And then probably most concerningly, there have been 3 human infections now that I know of. One in Missouri, well, 4 technically, because 2 in Missouri, one only documented by antibodies, but I think clearly a case. One case in a teenager in British Columbia in Canada, who's still in the ICU last I saw. And then one kid in California where the route of exposure was not known, did not at least have confirmed exposure to dairy animals or birds. And so that those unknown exposure cases are quite concerning.
yeah. Now, of the 55 or 57 cases that are reported, none of those have been documented to be transmitted to other humans. Does that indicate that the virus strain or clade that they had just doesn't have the right receptors yet to be transmitted from human to human? Because those dairy farm workers and poultry workers are right next to each other while they're working.
Right. So hard to say for sure. how somebody was infected, whether they were infected from a cow and milk, um, and all of the things that are done in a dairy parlor, or that they were infected by their colleague that they work with closely all day, who's also in the same environment. So, in general, these viruses, the current virus that's circulating, preferentially infects and binds to receptors that are found in birds and not people. And so far, we think the risk of human to human transmission is probably low because of that. But again, these viruses mutate. They are RNA viruses, just like COVID is, and we've seen how rapidly COVID mutates. Uh, Influenza viruses are similar. They do mutate, and so the concern is that mutation may eventually wind up with a virus that is capable of transmitting from person to person. But so far, we have no confirmed human to human transmission in this current outbreak. It doesn't mean it hasn't happened. But again, nothing that we can say with certainty, yes, that was definitely a human to human case.
One thing that was interesting for me as an ophthalmologist is apparently many of the dairy farm workers developed a conjunctivitis infection in their eyes. I guess it's from the
Yeah.
spray in the barn. It gets in their eyes, and the virus sticks to the surface of the eye, but for some reason doesn't get down into the lung from there.
Yeah, it again, we're learning a lot about influenza viruses and this virus even now. And that has been certainly one of the unique features of this outbreak. We've seen conjunctivitis in prior human infections with bird flu. It's more common than it is with seasonal flu, but certainly from this outbreak, it's been predominantly conjunctivitis more than any other symptom. And I think that probably is because of the route of exposure. And that may also have something to do with the severity of disease. We know that for many viruses and many pathogens, the route of exposure really impacts the progression of disease and your immune response. And so, one example is the smallpox virus, right? Variola virus. If you get the virus onto your mucous membranes, which is how it's normally transmitted from person to person, it creates a disease that has overall a case fatality ratio of 30%. If you take the virus, though, and you put it on a scratch or a scar or a wound on your skin, which was done to essentially the first vaccines of people were variolation, then it has a CFR of only about 1%, right? So just changing the route of infection for that virus significantly changes the course of disease. So I think the conjunctivitis thing may just be they're getting all of this splatter and droplets in the eyes from how dairy operations work. And that also may explain, at least in part, why we've seen much lower severity of these infections. Maybe that route of infection results in less severe disease.
Yeah, I've also read that the alpha receptor, which you mentioned earlier, is different on the conjunctiva than it is in the upper and lower respiratory tract. So
Right.
maybe these viruses just stick to that alpha receptor, and hopefully they won't mutate to uh, other alpha receptor. The Missouri case is interesting because nobody's been able to figure out how that person and the person they live with at home got it. They haven't had any contact with birds. They haven't had any contact with cows. So what's the current theory of how the Missouri couple got infected? And fortunately, they weren't that severely ill.
Right. Well, the first index case where the virus was actually detected was sick enough to be in the hospital. So this person was sick. Now they also had other comorbid conditions going on with this, so it's unclear how much of that was that how much of their illness was attributable to the flu or these other things that they had also. But I have not heard a good working theory of how uh, these two cohabitants were infected. And I think the lack of exposure to cows or dairy products or birds really means to me that the most likely source is potentially a person, right? So that they did have contact with some people, who came, from outside their household, and so if you're looking for a vector that brought the virus into the household, if you don't have an easy explanation of a cow or milk or a bird, you have to think a person is the next likely scenario.
Right. So they're in a grocery store shopping. The guy next to them is a dairy farm worker who sneezes, and
Who knows? Yes, right.
they get it. That's a possible theory. Now, you mentioned the California, the young child case. Apparently, that one, they think the child may have been exposed to some wild birds. So there's a possible explanation there. But the scariest case of all is this British Columbia, Canada case, where this is apparently a healthy teenager who didn't have any underlying conditions, gets the bird flu and has been in the ICU for 3 weeks on a respirator or ventilator, and apparently is still not out of the woods. And there's no evidence, again, of contact with birds or cows in this healthy teenager. So,
Right.
what's the best working theory there? Is it the same as the Missouri theory?
Again, I haven't heard a good working theory of how this teen might have been infected, and I obviously don't have all the details of the case. But if you've ruled out, um, a source from a cow or milk or a bird, then I think what's left as the most likely explanation is, again, it came from a person. And you can't confirm or positively attribute that until you have evidence and find somebody that they were a close contact of who has evidence of infection. But again, just by process of elimination, you have to think that it is certainly quite possible that uh, these folks acquired H5 from another person. And this wouldn't be the first time that that's happened, right?
Right.
We've had a number of documented person-to-person transmission episodes of H5 in Asia, um, but it is rare. And it's concerning because the more the virus has the opportunity to do that, and the more we see the virus do that, it's an indication that the virus is moving closer and closer to, again, a virus that can easily transmit from person to person. And once we've hit that threshold, again, an H5 virus that transmits easily from person to person, we're in a pandemic. And then it is 2020 all over again, maybe worse.
Now, for my small conspiracy theory audience, um, some people have mentioned to me that maybe somebody is purposely sprinkling some virus around and trying to infect people. Is that a crazy conspiracy theory?
Well, it is a conspiracy theory, and it does not have any basis in science. I'll leave the pejorative terms out, but we can trace these viruses pretty well using modern sequencing and genomic techniques. So we can essentially read the entire code of the virus genome, right? That's genomics using advanced sequencing, or even pieces of it. We can read the entire segment. And then we can look at how the virus has mutated over time and where it's moved. And so we can trace back where most of these infections came from. And so we know that these viruses, again, were introduced into cows. In one place, we know that cows infected other cows, cow to cow. And we know that the infections that came from poultry came from bird viruses that were circulating in bird populations. So all of this can really be mapped out using modern sequencing techniques. And so we would not see that type of diversity and evolutionary change that we've seen in this virus, uh, if it was all coming from the same, deliberate man-made source. So these infections in cows and the infections that we've seen in humans can be traced back, most of them to origins. And so you would then have to postulate that the few that we haven't been able to connect, like Missouri, for instance, are happening at the same time we're having these other large natural outbreaks, and that really just doesn't make much sense. I think we have very good explanations for how this virus is jumped around from birds into cows and into people in most cases, that I have, essentially zero concern that there's any deliberate, not even essentially, I have zero concern that
Sure.
there's any deliberate human efforts behind these infections.
All right, that's good to know. But in 2014, apparently there was some gain of function research done on bird flu viruses at the University of Wisconsin. Now, the government eventually banned that, but my understanding is in 2019 they allowed it again. So, is there gain of function research being done on bird flu viruses in different medical centers or universities around the country?
So that gain of function term has gained a lot of popularity here over the last couple of years, and I think that there's certainly legitimate concern about doing that type of research. And essentially, what that involves is taking a virus and manipulating the virus so that it potentially becomes more infectious to humans, or becomes more pathogenic in humans, right? Causes more severe disease. And that type of research needs to be done with close scrutiny and oversight. But also, that type of research is critically important for us to understand how do these viruses change so that they can adapt for humans, right? We wouldn't know about mutations that make these viruses more likely to transmit in humans and be able to track that unless people had done these experiments. It's also how we can develop new therapeutics and better vaccines for these viruses. Just like I talked about with Ebola, right? The reason we have now really exciting vaccines and potential therapeutics for Ebola, which is a horrible, highly fatal disease, is because we've done research in BSL-4 laboratories. And so this type of gain of function research, I would say, needs to be done under strict biosafety regimens. It needs to be done with very meticulous oversight. But it's also critically important to be able to protect us against these types of viruses. And there's no evidence that this influenza virus is related in any way to gain of function research or laboratory research that was done with influenza viruses. Again, we can trace this virus back to its lineages in the early 2000s. And so, I don't discount those concerns about gain of function research, in quotes. But I also would emphasize that type of research is important, and that we need to be able to develop, oversight and biosafety procedures that allow us to do that research safely.
Right. I think the key phrase you said is strict supervision of
Right.
what's going on. My understanding is some of these labs actually trade viruses with each other. I'm not sure how that's done, how the viruses are shipped, who handles them. So there's a
Right. Well, sharing of material and knowledge is as old as science, right? That's how science progresses. That's how, the scientific process and method works, right? We validate and we confirm what others have done, and then we build on that to be able to advance the science and the knowledge. So those, lots of different viruses and pathogens are shipped back and forth across the country all the time. For pathogens that are more dangerous, such as novel influenza viruses, there are strict procedures in place, in terms of tracking and safely moving those under the select agent program that's monitored and administered by the CDC. So there's rules about how you do those things. But again, if you don't allow that, if you outlaw all of those things, then the science can't move forward, and then we remain vulnerable to these viruses.
So, do you know how those packages are shipped? I mean, is it FedEx or UPS, or is it, under military guard while it's going from, Boston to Austin, Texas, or wherever?
Yeah, they are moved by commercial shippers, and they are packaged, with a variety of safety features built into it. So, honestly, you could throw the package off a building, and it wouldn't create a hazard to anybody. There were periods of time where folks in the biodefense community and the military biodefense community particularly wanted to have armed guards, running beside the vehicle with rifles. But, honestly, the safest way that many experts thought to ship these things were in, generic white packages buried in thousands of other generic white packages, rather than, the obvious, hi, we're shipping Ebola. Right. So, and again, all of these pathogens now, there's always been concern of, well, what if it falls into the wrong hands? Well, you can synthesize any virus in a laboratory now, right? You don't need to get your hands on the package that contains the Ebola virus or the bird flu virus. All you need to do, and actually most of these you can mail order from synthetic biology foundries.
Really?
Yeah, that's
So
been
I
shown.
can order some bird flu from some lab.
You can order elements, parts of it, and then assemble it yourself. There are safety triggers for certain sequences that, so if you tried to order smallpox virus, that should trigger, an alert that would go to authorities. But
Right.
there are lots of foundries in countries that don't have these safety provisions. And there was a very well done and somewhat scary publication a couple of years ago about a group that essentially assembled uh, horsepox virus by ordering pieces through the mail, and then assembling it in the laboratory. And horsepox is a very, very close relative of smallpox. So seems pretty easily feasible then that you could do the same thing for variola virus.
Hmm. Seems like a dangerous situation to me. All you need is a rogue UPS driver who recognizes the packaging of a dangerous virus and decides to do something bad with it. The analogy is what's going on with computer hackers. All you need is a rogue employee in some giant company, and they can destroy all of the computers or do some other nefarious things with them. So I don't know. To me, it sounds like there should be more controls on that. Uh, See what happens. Now, what about vaccines? Are the seasonal vaccines that we get every year that the hospital I work at forces me to take every year, are those of any value for the H5N1 virus?
Yeah, there's probably limited value from those vaccines in providing protection against H5. And again, that's why, a new influenza virus, with a new hemagglutinin or neuraminidase number is concerning because we don't have great immunity. There's some discussion about whether there's some similarities in the hemagglutinin receptors between H1 and H5 that may provide some crossover protection, but there's probably not much. And so there are currently H5 vaccines that are undergoing development and production. There were a number of H5 vaccine uh, starters, essentially H5 antigens that were developed and banked in the mid 2000s after those H5N1 outbreaks in Asia. The virus has kind of drifted away a bit, and so it's still unclear how effective those old vaccines will be against protecting for these new viruses. Those studies are being done now, but there are vaccines that are in development currently for this H5N1 virus.
Might that not be a little too late? Because potentially in 6 months, if the current trajectory continues, we could possibly have a pandemic in 6 months or so.
Right.
Um,
Well,
we going to have enough vaccine stockpile for that?
no, we do not have enough H5 vaccine stockpiled now to vaccinate a large proportion of Americans. There's again effort underway to develop new vaccines, including mRNA-based vaccines, which are much easier to rapidly scale up. As we saw with the COVID vaccines, the mRNA technology is really revolutionary in being able to scale up production quickly. The vaccines that are currently banked are kind of the traditionally grown and produced vaccines, either in old egg culture systems or in cell-based culture, but it still is, you have all of the idiosyncrasies of biology there in trying to produce um, large amounts of virus consistently in those systems.
What about Tamiflu? That's the traditional drug for treating patients with the standard seasonal flu. Is Tamiflu effective against H5N1? And I think there are a couple of other antivirals that work for the seasonal flu. They effective? Has that been tested?
Yes, so Tamiflu or oseltamivir should be effective against this H5N1 virus. It's been used to treat human cases of H5 before. The virus does not have mutations. The current virus that we've seen does not have mutations that would suggest that the efficacy of oseltamivir would be diminished. There are other neuraminidase inhibitors that can be used. Peramivir is one that's available IV. There are some older antiviral drugs, amantadine and rimantadine, which will not work against this virus. And actually, we don't use those very much anymore at all because our seasonal influenza viruses are mostly resistant to those drugs now. And then there's baloxavir, which is a new antiviral that has efficacy against influenza. So we do have antiviral treatments which work relatively well. Uh, just as the COVID antivirals, they work much better when they're delivered early. And so the earlier you start treatment, the more beneficial effect you get from those antivirals. And we do have a large stockpile of oseltamivir in the strategic national stockpile, millions of doses that are currently curated by the SNS to be able to respond to a pandemic. So
So
that's good.
we might need tens of millions of doses, though. If
Yes, indeed. And obviously, again, the critical piece is to get the antiviral drug into people early to prevent severe disease, also to potentially diminish spread from person to person. And the key or the trigger is to get it into somebody, you generally need to have a positive test. And so that whole chain of events of getting somebody to present with symptoms or at somebody who's had an exposure to get a test, to get the test result back in a rapid fashion so that you can administer the antiviral drug is one that we struggled with in COVID, and I expect we would struggle with in a flu pandemic as well.
do we have the testing, like a nasal swab, all ready to go for the H5N1, or
So influenza tests are used relatively commonly, and now many of them are the molecular-based tests as well as the antigen tests. The molecular-based tests, we should be able to swap out molecular tests for H5. Those are in development, but again, there's not a huge amount of investment to make those widely available now, so they're still only available in a limited number of laboratories. And antigen tests have significant limitations, and we don't have antigen tests currently available for H5. So right now, our diagnostic capability is limited. We essentially test for flu A. If it's flu A, we have many tests now that are able to subtype it immediately. So it'll tell you it's flu A and it's H1N1, or it's flu A and it's H3N2. But if you get a test result that says flu A, neither H1 or H3, then that needs to be sent to usually it's a state public health lab, which will have the ability to do additional testing to confirm H5.
Right. It's a catch-22 kind of, because as long as there's no pandemic, you don't want to spend billions of dollars creating all these tests and treatments if nothing ever happens. On the other hand, if it happens, you're not going to have the equipment and testing and drugs that you need. It's a difficult
Right.
situation.
Right. Well, there's certainly a lot of things that we could do in advance to prepare and to create a rapidly scalable system that you could then just plug and play with a new PCR assay that had already been developed and validated. But as generally happens, we will be back on our heels and until the actual event occurs, and then we will scramble and not be able to develop adequate testing and therapeutic delivery in time because again, as we saw early in COVID, these epidemics move incredibly quickly, right? It's a matter of weeks from the first cases of human transmission to potentially when you're seeing large outbreaks, particularly for flu, which has a doubling time that's generally even faster than COVID. So short incubation period, right? You quickly become infectious after becoming infected with the virus. So flu is a big challenge.
So clearly you're a major expert in this. What do you think, in your judgment, is the percentage chance that we're going to have a pandemic? Can you say?
Wow. I hate to give a percentage chance because I really don't have any.
Sorry to put you on the
I
spot,
know I
but
don't have
you're the
any
expert.
calculations to base it on. I would qualify it like this. I think the chances that this actually turns into a pandemic are pretty low. Uh,
That's good. Low
yeah.
means less than 50%.
Oh, well, yeah, much less than 50%, right?
All right,
But
that's great.
yeah. So the odds that it will, become a pandemic virus are very low. However, the consequences, if it does become a pandemic virus, are very high. And so to me, that translates into a risk that's not low and not negligible, right? So because if you have a low probability but high consequence of event, that is still potentially a significant risk. And that's where I think that, well, part of where our communication, I think, has really been poor about this whole event is, we keep saying, oh, it's low risk, don't worry. But you know
Right,
what? We should be a little worried. We should be worried about this virus and we should be doing things because when you say low risk, don't worry, it translates to people, including, people who make decisions about where money gets spent and resources are allocated. It means don't do anything. But that's the wrong answer, right? We should be running. We should be running fast. And again, that's one of the unfortunate things about public health is when public health works well, nothing happens. And so then people
right,
become complacent and wonder, well, why are we spending all this money on public health? There's no benefit from this, but the benefit is, yes, nothing happened. Um, and so we should be doing a lot. You really need to be like that duck on the water, right? The feet are paddling away wildly while you're gliding along the surface. But we're kind of like a dead duck that's floating upside down right now, and that's probably not the posture we want.
right. Yeah, it's interesting. When I go to all the websites of the CDC and I go to all the state government websites, they all say low risk, low risk, low risk. No, low
Yeah,
risk, don't worry
I
about
think that's the wrong message to send
it. Right. So they shouldn't
because
say
I don't
high
think it's
risk,
accurate.
but they should say there's a significant risk that
Correct.
this
Yeah.
could become a pandemic.
Yes. A concerning risk.
I like that. Now, as you know, one of the reasons that Trump probably lost his reelection in 2020 was the COVID thing. And do you think his administration now is getting ready for this? Kennedy is going to be the chief, and Dr. Oz is going to be there. Are they already working on this and setting things up to prepare for this? Do you know?
I really have no idea where pandemic and health emergency preparedness sits with certainly with Robert F. Kennedy Jr. or Dr. Oz. Some of the other candidates that have been named as potential positions, at FDA and CDC and other places, I think are very concerning because they have been voices that have undermined our COVID response. I think it's probably a safe prediction to say that pandemic preparedness is not going to be high on the priority list for a Trump
Mhm.
administration. But I certainly haven't heard a lot of discussion about H5N1 preparedness from any of the current named candidates.
Yeah, my other concern is when the new leaders go into their positions and offices, there's going to be a huge change of personnel. There's probably going to be people who quit, people get fired, they're going to hire new people who are loyal to them. And during that transition period, we may lose track of what's going on. But I don't know how to fix that.
Right. I think there's certainly a potential for chaos. One of the good things about you know one of the good things about our federal government and system is things change slowly. Sometimes that's a really bad thing,
Right.
but sometimes I mean the system was not built to be a rapidly movable vehicle, right? It's more like a battleship. Things change slowly. And so while the political appointee level, which is assistant secretary and above in departments and agencies will turn over, everybody else below that, all of the career civil servants and government workers will stay the same. And there's some benefit in that, right? There's incredible institutional knowledge. There's technical expertise. There's also, bureaucracy and inefficiency and all of those things we know about. But there is, there is a lot of uh, optimistic potential for, the career civil servants, which have been doing this for a long time. And again, and in some departments and agencies that deal with these issues have some of the, really best minds and experts. And so hopefully those folks will remain in place and will continue to do their jobs.
And I think they need to hire you to stay on top of this because
I,
you have a significant concern here, which I like to see.
I can tell you the probability of that is much lower than of H5 becoming a pandemic.
You never know after they watch this podcast. I'm not Joe Rogan yet, but after they watch this, they may say, we need to get Lawler in here to tell us what to do next. So
Yeah,
now, I interviewed Dr. Peter Polisi about 7 months ago on bird flu. The situation was much different back then. There were just a few cows and chickens infected. It didn't look too serious. And he was optimistic that things would be okay. But we talked a little bit about COVID as well. And towards the end of the podcast, I asked Dr. Polisi, who was one of the top flu virologists on the planet. I said, by the way, with regard to COVID, do you think that was a lab leak from the Wuhan lab or not? And he said, I take the Fifth Amendment. What do you think? You're knowledgeable and experienced about this. What do you think happened there? Or do you want to take the Fifth Amendment as well?
no, I won't. I won't take the fifth. Although I am nowhere in the league of Dr. Palese in terms of my virology knowledge. But I think the reality is we're never going to know for sure where SARS-CoV-2 originated and how it got into people. I think it's unfortunately remote enough that many of the clues that could have been useful are gone. I think many of them may have been intentionally erased, because I think that to be honest, neither of those competing theories, the two most likely that it naturally emerged out of, a wet market in Wuhan into people, or it was the result of a lab leak. Neither of those are really necessarily appealing to the Chinese government. Neither makes China look very good. And
Right.
so um, I'm not sure we're going to be able to find the evidence we need to make a definitive judgment. I think the important thing for people to acknowledge, and on all sides. And again, this has become, unfortunately, a very politicized and ideological issue that's become almost uh, religious canon rather than just looking at facts. And to me, the important thing to acknowledge is that either theory is really plausible from the data we have, and it is very it is foolish and unwise and dangerous to discount one or another just because it doesn't align with our political ideology or the way we think the world works. The evidence, clearly supports either, and that means we should take steps to prevent either event from happening again in the future. And I think that's, as much as we're going to be able to take away from it. I can easily make the argument for either, and based on, rational and sound scientific data.
Hmm. It's an interesting observation. I haven't heard that before. It's usually people on one side or the other saying the other side is stupid or crazy or liars. But it,
Right.
the evidence, unfortunately, is gone. I think you're right. So we'll never be able to say with 100% certainty.
Yes, unfortunately.
Right. Well, I want to thank you again very much for taking your time to come on the program today and discuss this extremely important topic. This was very educational and informative for me, and I'm sure people out there watching this and listening to this will also appreciate it. So thank you very much.
Thank you. I enjoyed being on the program and look forward to speaking with you again.
Great, great. Thank you.